Our last speaker in this session is Professor Manish Raizada. Dr. Raizada is Associate Professor at the International Relations Officer in the Department of Plant Agriculture at the University of Guelph in Ontario. Professor Raizada received his Ph.D. from the University of Western Ontario in genetics and his Ph.D. from Stanford University in plant molecular genetics. The focus of the Raizada Lab is to develop low-cost technology that reduce the use of synthetic nitrogen fertilizer and pesticide to grow corn and other crops, replacing them with more natural biological resources with a focus on North America, Sub-Saharan Africa, and South Asia. Dr. Raizada is founder of SACS Global, SACS Sustainable Agricultural Kit, an effort to bring $1 technology to the world, 1 billion subsistence farmers who earn $1 per day. SAC kits are based on the principle of sustainable ecological agriculture. The presentation of Professor Raizada is improvement in biological nitrogen fixation in legume.
Quick propaganda slide about the University of Guelph. It's Canada's oldest agricultural university, and the rest is propaganda. It's located near Toronto. I'm going to talk a lot about work of Hanan Shad, a Ph.D. student in my lab, and Melinda, a postdoc in my lab. So today I'm going to talk about optimizing nitrogen fixation in legumes, helping farmers to overcome barriers to maximizing legume production, and our SAC strategy. So let's start with the first, optimizing symbiotic nitrogen fixation. So I need to talk about a quick introduction. So globally, 75% or so of malnutrition in rural areas is in rural areas, of which amino acid deficiency is especially problematic. Nitrogen fertilizers are also expensive. So in the case of symbiotic nitrogen fixation, as you mostly know, rhizobia bacteria inside legume roots, nodules convert atmospheric nitrogen gas into ammonia to build protein and other organic molecules. Now, how can nitrogen fixation be improved to help farmers, especially smallholder farmers? Well, there's some problems. One could be the incorrect native rhizobia in the soil. So a solution to that would be to coat seeds, for example, and those are called inoculants. Problem two could be a poor crop variety, so you can breed or select for legumes with improved nitrogen fixation. The third problem could be low micronutrients in soil, such especially molybdenum and boron, or other limiting macronutrients such as phosphorus. Now, for all of these, one needs to diagnose the problem and test different possible solutions. So, for example, test different rhizobia strains. Current methods for assessing symbiotic nitrogen fixation have limitations. I won't go through them. The gold standard is considered the isotope method or N15 method. They can vary in reliability, they're time-consuming, they can be expensive. Some of these are $10 to $20 per sample. The isotope method is a very good method. Like I said, it's the gold standard. It is a bit expensive, though. So we need an efficient, low-cost method to measure SNF, especially for developing nations, to help smallholder farmers. So as background here, it's important to know that in legumes, a portion, either a small or large portion of fixed nitrogen, is transferred to leaves from the nodules as amino acids, such as glutamine. Okay, so that's some background. So now, how are we trying to tackle part of this problem? So I need to introduce you to this concept of a biosensor. In this case, we've engineered a very simple biosensor in E. coli. So what it is, this is an auxotroph. In other words, it cannot grow in the absence of a particular metabolite. In this case, that's the amino acid glutamine. So it detects the amino acid glutamine, and in response, so what happens here is that in its bacterial chromosome, it has a mutation in a gene called glutamine synthetase. It cannot make its own glutamine. When you supply it with exogenous glutamine, the cell starts to grow. And in this case, we've engineered it so that it emits photons of light. Okay, so how it works here is that imagine if you have a nodule here. In the nodule, N2 gas is being converted into ammonium, which can be secreted as ammonium. In some situations, it can be secreted as glutamine. We then incubate either the tissue or rhizobia cells with our biosensor, and we measure photons of light. It sounds complicated, but actually it's quite easy. So I'll show you a couple of assays. Let me describe one assay. So In this assay, which we call the Glutamine Lux Agar assay, it's a method for high-throughput screening of bacterial colonies for nitrogen fixation output, and it's a 1 to 2 hour protocol. So in this assay, what happens is that we embed biosensor cells in agar, and that's what those black dots are. We then put microbial colonies, these could be rhizobial colonies, shown in orange, on top of the agar. We let that incubate. If it's fixing nitrogen, there should be some release of amino acids, including glutamine. We then flip the plate upside down, and we look for photon emission from the adjacent biosensor. Okay, so does it work? The answer is yes. So what I'm showing you here, this is in vitro now. These are colonies of, the first is a Bradyrhizobium japonicum strain, so it nodulates soybean. And what you see for validation purposes is the wild type strain, and this is a single gene mutation that cannot fix nitrogen. So you can see our biosensor actually very rapidly, within 8 hours, is picking up a difference. This is at 22 hours. This is a different strain. This is Cynorhizobium mellilodi that nodulates alfalfa. So what we have then is a method for picking up in vitro nitrogen fixation. I'm going to skip that for time's sakes, but what this assay permits is high-throughput screening of in vitro nitrogen fixation. What we can actually do now is screen thousands of colonies per day. There is some capital cost associated with it, but it's actually a relatively simple assay. So it permits screening of new nitrogen-fixing bacteria and potentially selection for improved improved stress tolerance. We're using this technology not only for rhizobia, but for other microbes. So what I'm showing you here is the same type of assay, but this is 96 in 96-well format. These are 96 microbes that we isolated from corn seeds. So these inhabit corn seeds. They're known as endophytes, which is actually a large focus of my lab. And in this case, you can see that out of this plate, this actually had been selected on low nitrogen media, so it's biased. We picked up a lot of nitrogen-fixing endophytes, and we validated it using 2 other methods, a cellulose reduction assay and dot blot hybridization with a NIF probe. Okay. Now, so we have a technology on the in vitro microbial side, but we also have used this to look at in planta nitrogen fixation. So in this case, what the goal here is to be able to look at nitrogen fixation output at nodule scale resolution. So what we can do is we can take an intact root system of a legume, freeze thaw it. We freeze thaw it to cause glutamine leakage. The cell walls break open. We put those root systems onto agar. The agar contains our biosensor. Glutamine leaks out, and then our biosensor picks it up on a CCD camera. So I'm going to show you some experiments here. These are in growth pouches. The important thing to note here is that there's very little starter nitrogen in the background. Okay, so on the top, so these are split root experiments for validation purposes. What's happening is half the root system was inoculated with a wild type rhizobia strain. The other half was with the single gene mutation, so it cannot fix nitrogen, but it can nodulate. And so what you see here, the top panel is our green peas, and if you look on the right here, see these red dots? Those are actively, we think those are actively fixing nodules. Soybean, same thing. Okay. Alfalfa, same thing on the right, and lentil, same thing. Okay. So that gives us nodule scale resolution, and the nice thing about that is now we're using it to look at some questions. So, for example, it hasn't been clear how much nodule-to-nodule variation there is for nitrogen fixation. So, for example, if you put something under drought stress, lots of nitrogen in the soil, other types of stress, pH, does it affect all nodules equally? Does it affect some nodules specifically in certain parts of the root system? So this sort of nodule scale resolution can help decipher that. But the assay that's particularly useful in a smallholder context is this one. The others are particularly useful for basic research, I think. In this assay, it uses a single leaf punch, okay, to look at nitrogen fixation output, and again, glutamine. So this is a 3-hour protocol, and the cost in Canada is about $1 per sample. We've transferred the technology to Nepal. It's about 10 cents per sample there because of labor costs. So what we do here is we can take a single leaf punch. Now, these are all plants grown under controlled conditions, not outdoors. I'll get to that in a second. We take a single leaf punch, we freeze thaw it, we grind it, we take the supernatant to get glutamine. We could take 96 punches at a time, put them in 96 well plates, add the biosensor to it, let it incubate for a few hours, and then in a 96 well luminometer, which measures photon output, we look at how much glutamine indirectly was there. So, for example, in lentil, so the experiment here is what is the effect, can this technology decipher the efficacy of different rhizobia inoculants? So this is the effect of different rhizobia strains on symbiotic nitrogen fixation in lentil. So what I'm showing you here is this was uninoculated. These are inoculated with different strains. This is the leaf punch, and we always take a leaf punch from a growing leaf because that's where free glutamine will be migrating as a sink. And you can see that we have variation here. Okay? Now, I think I'm missing some. There it is. Okay. If you look at the gold standard, which is the isotope, the N15 isotope assay, so this is nitrogen derived from the atmosphere, you can see it's pretty, correlates pretty well. And the correlation is right here. The R squared between, sorry, that's not the right one. The R squared is actually almost perfect in this case. So it's glutamine lux leaf punch assay, which is less than $1 per sample, versus this method, which is $10 to $20 per sample. Under controlled conditions, it correlates very nicely. And of course, that does make sense because the only nitrogen that's coming is from nitrogen fixation, except for that in the seed. I'll skip this, but we can image it. In common bean, same sort of, now in the common bean, I'm just showing you a different sort of experiment. And the question here is, could we distinguish between different varieties of bean for their ability to fix nitrogen? And I had a colleague who had already investigated this. So here's our glutamine lux result. Here is nitrogen derived from the atmosphere. The y-axis is slightly different because the biomass changed, so we had to multiply this by biomass. This was a non-nodulating strain, so that was used as a comparison. That's why it's out here. But you can see the rank order is not bad. And here's the relevant metric. It's not bad. There's some variation, but the R-squared is pretty good. So the technology, this cheap technology can pick up both varietal differences as well as rhizobial inoculant differences, at least under current controlled conditions. I'll skip that. Now, is there any relevance to the field? Okay, so this was the data I think I was just showing you. So these are different common bean varieties. They vary in nitrogen fixation output. And you can see it here. This is our leaf punch assay. These are the same varieties grown in the field, in a low nitrogen field. That's actually quite good. This actually surprised us. We were not expecting this. And this is nitrogen derived from the atmosphere. Again, R99 is non-fixing. That's why it's not shown there. So we have some hope that under field conditions, at least under low nitrogen field conditions, this may be informative. So we know that at least under controlled conditions, this is very good. However, and that can of course help smallholder farmers as an early screen. So for example, if in a breeding program, one wants to know, how if you want to set up a matrix of different crop varieties and different rhizobia and say, okay, which appears to be the top 10 candidates, top 10 combinations, this may be useful, okay? Now, the next question is far more difficult, and that is, can we use this to diagnose problems in the field, so suboptimal nitrogen fixation in the field? So why not just do the most challenging experiment possible, which is go directly onto terrace farms in Nepal, where we have a project, and do farmer trials. So we are now doing extensive farmer trials, and as I said earlier, we've transferred this technology to Nepal to an NGO partner. What I can say first is that this is adjusted grain yield, okay, and this is farmer number 22. And if this is the traditional farmer practice, uninoculated. If you add micronutrients, if you add local rhizobia, you can see, it's 2.5-fold increase. But somewhere nearby, on a different plot, you can see that adding rhizobia actually causes a decrease. That's for common bean. Cowpea, same sort of thing. This causes a slight decrease, this causes an increase. And that's because, of course, there's tremendous plot-to-plot variation in terms of background rhizobia, in terms of nutrients, et cetera. The reason I'm bringing this up in part is to say that the rhizobia response, just like the nitrogen response, is site-specific. And hence, a site-specific technology is needed, whether it's glutamine lux or something else. So if we're going to actually have a practical tool in the field to tell this farmer you should add this rhizobia strain or not, we need something that's very inexpensive. I don't know if this will get us there or not. Okay, right now we're awaiting the first-year field results. Actually, we have some of the results, just some of them have not been analyzed yet. But clearly, there's going to be site-to-site variation. So that's what we're trying to do. So just to conclude this part of the talk, it's a new method to measure nitrogen fixation output. By the way, it works in the two types of legumes that are out there. We can use single leaf punches. It's rapid, low cost, high throughput. The Agar assay permits visualization of active sites. We think we can use it, well, as a pre-screen, both for varieties and strains. And the Agar assay permits screening of thousands of colonies rapidly. It is only measuring glutamine, which limits the assay, which I'm happy to chat about. Okay, I'm going to switch slightly here. I look at this problem of maximizing nitrogen fixation a little bit more holistically. So helping farmers to overcome barriers to maximize legume production, which in turn will fix nitrogen, is part of a larger project that we have in Nepal. It's part of a $2 million grant funded by the Canadian International Development Research Centre, IDRC, and Global Affairs Canada. So it's focused on terrace farms. Now, I'm going to show you some pictures that we've developed that we're using as teaching and extension tools in Nepal. But I'm going to take you through this kind of quickly. So in the box here is for example, sometimes is a traditional practice or is what the problem is. So what we're trying to tell farmers here who may not know much about nodules is, okay, if you want to self-diagnose the problem, take a nodule from a legume, cut it open, use a magnifying glass, and some of the products as I'll show you later, some of the products that we sell are magnifying glasses, okay? And I'll show you the multiple uses of a magnifying glass. And you'll see this comes from the dollar store, okay? Because there's lots of products, and that costs $1.25 here in Canada. Of course, these are produced in China, so the shipping cost in Nepal is much cheaper, right? Okay, so what we say is look at the nodules. Okay, if it's yellow inside, if it's not pink, it may not be actively fixing nitrogen. So that might mean you may have to supplement with nitrogen fertilizer, okay? If, however, you cut it open and it's pink inside, you're in good shape. Okay, you don't need to purchase nitrogen fertilizer, okay? So that's something we can teach in picture form. And why picture form? Because who we're trying to target are women farmers, many of whom in this context are illiterate. Now, we've been doing a number of field trials to try to intensify production. Some were mentioned earlier. I'm just showing you one of the 8 or 9, both rotation and intercrop trials we're doing. So this one is sowing maize together with cowpea will yield more profit than maize only. We know this from our field trials. So here's maize only. Okay, you get one pile of grain there. But if you, in this case, mixed intercrop with cowpea, you get one of cowpea, one of maize. If you row intercrop, so alternating rows in lines, you get better improvement, okay? That's because you can walk between the rows, you can look for diseases, you can have better cultural management. Okay, I think I can show you some of our data here. I'm going to go through this quickly, but basically, in terms of total yield increase, we can achieve increases of 50% in grain, 85% in biomass, okay? Which then, of course, adds to soil organic matter. And those are pictures from some of our trials. Now, if you're going to do row intercropping, how are you going to do that? Especially on a terrace condition, right? It's not like they've got tractors to do this. So in this case, then, again, this is very holistic. You have to think about the whole farmer and the whole system, is we then introduce, actually we sell, jab planters. Oh, sorry. So this, if you've never seen a jab planter before in the remote hills of Nepal, like what is this thing, right? If you go to a store and try to buy it. So we have to explain what it is. So here's the traditional practice. It requires 2 people. So here's a jab planter, and you only need 1 person to do it, and you can plant in rows, and you get better yield increase. This, by the way, is important because in many parts of the world, men out-migrate to cities or even to other countries for labor, and so women are stuck doing a lot of the labor. So this is very important. And there's taboos around the world to using livestock. That's why the man is driving the livestock here, okay? That's why this tool is important. This is about a $20 tool, which is more expensive than I normally like. Once it's produced locally in Nepal, the price will come down. Okay, so here's traditional practice. Here's a woman using this particular model, but we have 3 models. Do farmers like this? Okay, well, here's preference with respect to tiredness. So in this, we work with test farmers, so 35 said they prefer the jab planter, 6 plow, 1 was indifferent. Would you use this in the future? Yes. Okay, in terms of intensifying agriculture, one of the problems in terrace farms is a lack of surface area. But there's a lot of vertical surface area that's largely underutilized. So one of the experiments that we've been doing, and I should also say at the edges, so they grow corn on these terraces like the Incas did. But at the edges, maize yields very poorly, okay? And so then you also get soil erosion off the edges. So we're trying different experiments of different climbing crops that climb up the wall. We plant it at the base of the wall. This, of course, are climbing beans. That's not doing so well probably because of shading issues. Other crops, I'll tell you, do exceptionally well. Coyote, squash, the curcurbite family does exceptionally well. Now here's the opposite, which is planting at the edge, the top edge, and letting it grow down like a waterfall. So these are trailing type crops. So these do very, very well. So black gram, rice bean, kidney bean, cowpea. Some farmers were already practicing this. We learned the innovation from them, actually, and now we're trying to spread it. How does this do? So from planting at the edge with these legumes, here's the net benefit of doing that, okay? So this is extra income now to the family. This is not inconsiderable. Here's some pictures from the trials at the edges. The dry season is very critical around the world. When we talk about 800 million people being chronically malnourished in rural areas around the world, a lot of that is seasonal hunger during the dry season. So one of the things that happens is during the dry season is that livestock have no food. So vetch is a feed that's nitrogen fixing. It's very drought tolerant. It can be planted under residual moisture. So what this is showing is traditionally, there's nothing in the dry season. On the onset of rains, the soil has cracked. You get tremendous erosion and the livestock are hungry. Plant vetch on residual rain before the dry season starts. It'll cover the soils and you have some livestock fodder. We're actually doing experiments trying to select for more drought tolerant vetch accessions right now. Okay, so that all sounds reasonable, right? Now I'm going to go into crazy territory. Again, you have to treat this holistically. Women do a lot of the work. Women, what are barriers to production and what are barriers to happiness? Well, women spend a lot of their time on their knees in the field. So look what, these are knee pads. These cost about 50 cents locally, okay? This is one of our lead products that we're selling. So traditionally, women, well, I think you got the point. Gloves. These are things, I'm a molecular biologist, right? I focus on high-tech stuff. But then we forget the little things. Gloves, right? We take this for advantage. They have gardening gloves. No, they don't. They've never seen this before. Okay, so we're trying 3 styles of this. Very cheap. Again, this is from a dollar store, $1.25, okay? We have different versions of this. And yes, the women like particular colors, okay? That actually is important, right? It's important for us, too. I talked about the magnifying glass earlier. So the best thing you could do, of course, is make sure that your planting material, whether it's corn or legumes, it's clean of disease, right? So farmers know this. I mean, they're always sorting seeds. But if you have a small grain legume like a small grain cereal like finger millet or a small legume, which sometimes are the most nutritious, you can't see small spots on it. So what do we introduce? A magnifying glass or a magnifying sheet. And that helps with seed sorting, okay? Again, a very inexpensive technology. Just 5-fold magnification is required. And that's what this is trying to say. Another technology is called seed flotation. So the traditional practice is you might take everything, plant it, and you'll get variable yields because some of these are sick. Some of the seeds may have been eaten by pests, and that means you're transferring it to the field upon planting. This is a beautiful technology, very simple. You in a jar, you add some water. You might have to add some salt to buoyancy, for buoyancy. You add your seeds in here. The lightweight seeds that have been damaged, right, by fungi or insects, they will float on top. If you were to plant those, you'll have a terrible field. But you just remove them, plant the heavy seeds, and you get a nicer field, right? This is a free technology. You just have to tell people about it, okay? Which is how we're scaling up using these pictures. Weeds. It's criminal in the 20th century that women spend half their time on the ground pulling weeds by hand. It amounts to, I've forgotten the exact figure. In Africa, it's 100 million hours per year, I believe is the figure, okay? It's criminal. So there's different approaches to this. One are cover crops. I'm not going to show you that here. The other are different tool designs. And the reason for these pictures is to try to inspire local blacksmiths. I'm sorry, I've lost that. there. Try to inspire local blacksmiths to, to produce various types of tools. This, by the way, is an indigenous Indian innovation. This is a stick with nails in it. Okay, that's all it is, all right? And it works pretty well. Okay, local blacksmiths on our project, using participatory methods, they like this design, so this is now being produced locally as a weeding tool, actually multifunction tool. Next is something you may have heard about, which is a grain storage bag. This costs about $1.25. It's produced by a company called Grain Pro. You may know about the other bag as well, called the PIX, Purdue Improved Cowpea Storage Bag. So the idea here is that about, depending on where it is, maybe up to 50% of the harvest can be lost due to insects and fungi, et cetera. And if you were to plant some of these seeds, you'd get a terrible yield. Here, what you do is make sure you dry the seeds properly. You put them in a bag. These bags are hermetic bags. What happens is oxygen flows out but not back in. Without oxygen, fungi and insects cannot survive. So you store this, you put this in a secondary container. If you were to use these seeds, you get good yield. My lab does a lot of work on microbes that suppress Fusarium disease, which produces mycotoxins. In sub-Saharan Africa, a little bit in South Asia, you get tremendous mycotoxin production. If you can, and that some of that happens during storage. So if you can suppress the fungal agents that create these mycotoxins, you can also have a health benefit in terms of, for example, reducing cancer rates. That particularly is the case in sub-Saharan Africa. A couple more things. So someone mentioned markets earlier. Look, if you buy a loaf of bread here, I think this figure still holds true. Let's say you buy a loaf of bread for $2, only 14 cents goes to the farmer, right? The rest is who bakes the bread, mills the flour, sells it, et cetera. So the profit is after the farm. In developing countries, they don't catch this profit. So this little lesson, that we have a few lessons on post-harvest value addition. This lesson is saying, okay, grow peanuts, wonderful legume. Don't sell it in bulk like this. Put it in a bag, put a nice label on it. We're suckers for that. You know how much extra we pay because something looks beautiful? They can double or triple what they get in income because of this, okay? So a couple of, I think this is the last slide on this part of it. Nutritionally, stunting is a major problem, for example, in South Asia. It's a major problem, and part of that is due to amino acid deficiencies. A lack of certain amino acids, lysine, tryptophan, methionine, causes during pregnancy causes low birth weight babies, or in early children causes subsequent stunting and some other issues. So this little lesson is trying to say, look, at meals, don't just eat corn, rice, cassava, etc. Make sure there's some legumes in here, and that will prevent stunting, okay? Okay, last part of my talk. How am I doing on time? 0? Okay, I'll be very quick. Okay. So I keep talking about the Sustainable Agriculture Kit project. What is it? It's a challenge. It's trying to address a challenge of scaling up that's already been talked about. So there's 400 million smallholder farms that have less than 2 hectares of land, and they have very little cash income. They live in remote areas, have very little knowledge extension support, poor access to private sector inputs and markets. And many of projects are not scaled up to impact these 2 billion or so people, right? A family of 5 times 400 million farms. Now, the challenge of scaling up is enormous in a place like Nepal, in mountainous areas. So here's our methodology, and I'm going to have to go through this very fast. Step 1 is to survey local farmers for their needs and innovations in partnership with a grassroots NGO. So we did that. Here is one question that we asked. Basically, how do you harvest your corn, or what are problems you have? And farmers said, look, women said this is a big problem. How they take kernels off of cobs, if they put those cobs in a sack and they beat them with a stick. First of all, that causes grain damage, which then leads to mycotoxins, right? Okay, there's different sites. So a proposed intervention was, I'm a corn geneticist, so here's the traditional practice, was a little hand sheller. This is $2. Well, locally it's $2, okay? You twist it on a cob of corn. Any corn geneticist here knows this tool. Very few subsistence farmers have ever heard of this, okay? So what's the impact of that? I'll show you the impact later. I'll do that very quickly. Now, decide a menu of innovations based on what the farmers are telling you of their needs that are low cost, purchasable by them, low labor, women-friendly, and sustainable. So it's a menu. Just like a restaurant menu, we have a menu. And farmers, and here's our current menu. Well, here's our menu from last year. So farmers can purchase whatever they want from this menu. Some of these are practices, okay, to meet their particular needs. So that's what I mean by a kit. There's not actually a kit, it's a menu. And people can buy whatever they want from that menu. What we then do, in our case with a group of 600 test farmers, is we test these innovations across 2 seasons. So for example, going back to the corn sheller. Okay, do you like this? Yes, they love this corn sheller. Our female involvement was very good. So women say things like, I can remove 80 kilograms of grain in a single day. They love this thing. $2. It saves about 2 weeks of hard labor. Okay? We've already procured 11,000 of these. Another 15,000 more are planned. Okay? $2 technology. Anyone can afford $2 around the world. Okay? Or most people can. So these are a long list of our demonstrations that we do. Again, it involves about 660 test farmers. Next step is use participatory surveys with the test farmers to rank the best innovations for scaling up, to prioritize that. So here's an exercise, small number of farmers, okay? So they were asked questions like basically rank it on various criteria, okay? So these were the top scorers, okay? I'm obviously not describing most of these to you. Then we know what to focus in on. Next, someone mentioned, our colleague mentioned distribution. This is how we deal with the problem, is to procure and sell individual items from the regional menu to what we call consumer farmers using pre-existing snack food, cigarette, and alcohol dealers directly into village stalls, okay, using a spin-off company from our NGO partner, okay, which is why we chose them. Anywhere I've been in the world, whether it's Central Africa, remote mountainous areas of Nepal, I could buy Marlboro cigarettes if I wanted. I could buy vodka. That's what that is, vodka. This is in the mid-hills of Nepal. I could buy Smirnoff vodka. So that means there are distribution networks. We don't need to invent them. We just piggyback on top of them, and that's what we're doing. So these are our current private sector partners. So these are peri-urban snack food dealers from where these distributors go into villages. So currently we have 21 of them. Small machinery dealers in peri-urban areas, obviously farmer cooperatives, traditional shops. So right now we're working with 104 dealers. Step 6 is to accompany each product with instructions in picture format. And a menu of practices in picture booklets. And I've already showed you some of these. So in the case of the corn sheller, here's the lesson. Here's the traditional way of beating it with a stick. Use a corn sheller, right? So this is also used as a poster for marketing purposes. This accompanies the product. So here's the book. So that accompanies the product. But when we put all this together, there's a large book now that's been created. It's 190 pages, 150 lessons. Individual lessons accompany products, but smaller customized farmer booklets are available. This is all free. Okay, I've made this all free and open access. I've worked with an incredibly talented graphic design artist. This is the South Asian version. This is, of course, they can't read English, so this is translated into Nepali, okay, the entire book. We did something called participatory editing with women farmers, and they requested 500 edits, okay? There was no text captions. So it was as if they were illiterate, and they said, I don't understand what that is. What is that crop, right? What are you trying to say? 500 edits. It took actually almost 6 months just to deal with the edits. There's other versions. So the African version is now complete. Lisa's now working on versions for Southeast Asia, Latin America. That shouldn't be North America. That should be North Africa and Middle East. Okay. Users can download individual lessons, add their own text translations, and create custom booklets. Lastly, is we use mobile phones to obtain feedback from consumer farmers on the efficacy and improvements needed. The cell phone penetration rate in Nepal is 83%. We've put in a dealer incentive so that when someone buys a product to record their name and cell phone number. So our goal as part of this project is to reach 100,000 people in the next 2 years, 25,000 households. With some of our lead products, we're already well on our way to that. So this is what I've told you about today. A laboratory-based test for improving nitrogen fixation, looking at barriers to maximizing legume production very holistically, and then our SAC strategy. And here's our website. I didn't put the Twitter handle here, but we have 7,500 Twitter followers at this point. The SAC picture book is going to be put online next month. It's actually, you can link from it from here, but it's actually sacbook.org. Thank you for your patience.